discuss some of the biochemical tests that are frequently employed in the microbiology lab. Indole test. Indole test is performed to determine the ability of the organism to split tryptophan molecule into indole. Indole is one of the metabolic degradation product of amino acid tryptophan. Bacteria that possess the enzyme tryptophanase are capable of hydrolyzing and deaminating tryptophan with the production of indole, pyruvic acid and ammonia. Why do we perform indole test? We perform indole test because it helps us to differentiate species of the family Enterobacteriaceae. The procedure is written over here, but it is not important for the exam point of view in UHS. But indole positive and indole negative are important. Those members of the Enterobacteriaceae family that are indole positive include E. coli and Proteus vulgaris. And those that are indole negative include Salmonella species, Klebsiella species and Enterobacter aerogens. Citrate utilization test. This test is one of the several techniques used to assist in the identification of Enterobacteriaceae family. This test is based on the ability of an organism to use citrate as its only sole source of carbon and ammonia as its only source of nitrogen. Principle. The test organism is cultured in a medium which contains sodium citrate, an ammonium salt and the indicator bromothymol blue. Growth in the medium is shown by turbidity and a change in color of the indicator from light green to blue due to alkaline reaction following citrate utilization. It is positive in case of Klebsiella pneumoniae and negative in case of E. coli. TSI, triple sugar iron test. It is used to determine whether a gram-negative rod utilizes glucose and lactose or sucrose fermentatively and forms hydrogen sulfide. The formation of carbon dioxide and hydrogen is indicated by the presence of bubbles or cracks in the agar or by separation of the agar from the sides or bottom of the tube. The production of hydrogen sulfide requires an acidic environment and is indicated by blackening of the butt of the medium in the tube. Now the result interpretation. This is called the butt and this is called the slant. Alkaline slant and no change in the butt occurs in case of glucose, lactose, sucrose, non-utilizers. Alkaline slant and acid butt occurs in glucose fermentation only. Here is an acidic butt, an alkaline slant. Acid slant and acid butt with gas production. Glucose, sucrose and or lactose fermenters. This one. Alkaline slant and acid butt, H2S production, glucose fermentation only. This one. So, I will repeat this result interpretation. You can see in this tube there is no change in color, so we can say that its slant is also alkaline and there is no change in the butt. So over here there are some bacteria that are not utilizing glucose, lactose and sucrose. No glucose, no lactose and no sucrose is being utilized. In this one you can see that the butt is blackish in color and the slant has the same color. So in this case there is glucose fermentation only and there is H2S production. In this one you can see that there is alkaline slant and the butt is yellowish in color, so it is acidic. This shows glucose fermentation but without H2S production. And in the first tube you can see that the whole thing, the butt as well as the slant has turned yellow. So everything is acidic now and there is production of gas. This shows that there is a glucose, sucrose and lactose fermenter. The first one with acid slant and acid butt and having a gas will be E. coli. This one who has got an alkaline slant with an acidic butt having H2S production is Salmonella typhi. In the next section, we will study the test for enzymes.